Hello again, welcome back. So today we're going to talk about making representations in the brain. This is the first step to cognition. Cognition is often characterized as information processing, so you can say that cognition starts as soon as information gets in the brain. How does information get in the brain? It's through our perceptual systems. So perception is the first step in cognition. How does perception happen? Well, in two stages. The first thing is we have a sensation. Something out in the world starts to impinge upon our sensors, our visual sensors or our hearing sensors or our touch sensors, and we have a sensation. This is then integrated into a percept, which we then, in a second stage, with further processing, can recognize and characterize perhaps as something we've already experienced before. How do we go about doing this? Well, there are two views. One is a very direct view. It was proposed by J.J. Gibbons way back in the 1960s. This view is quite simple. And what he said was, well, whatever's out there gets perceived. So if you have a stimulus, if there's light in the world and we have working visual sensors, we will see light. That's a very direct view of seeing whatever is out there in the world. An alternate view is that of Hermann von Helmholtz. He proposed it back in the 1800s, actually. And this says that we actually are involved in perception and we often perceive a little more or a little differently from what's out there in the world. Helmholtz proposed the idea of unconscious inferences affecting our perceptions. So let's take the example of visual perception and see how both of these play a role in perception. We know that people are equipped with senses such as sight, hearing, and taste that help us take in the world, right? Amazingly though, these senses have the ability to convert real-world information into electrical information that can be processed by our brains. The way we interpret this information, our perceptions, is what leads to our experiences of the world. In this module, you will learn about biological processes of sensation and how these can be combined to create perceptions. We will use the visual system as an example. It's the modality through which a lot of our knowledge is acquired and interpreted. And vision provides an excellent illustration of how the close study of the brain can proceed and what it can teach us. Here is a good example of a scene. It's a fairly cluttered scene. You might recognize it as something from a Where's Waldo kind of situation. And you might find yourself already looking for Waldo in this picture. Think about what attributes it is that you are looking for. What features, what aspects of each individual are you considering when you decide if that person is Waldo or not? Have you found him yet? Don't worry if you haven't. Clearly then, features are an important aspect of perception. However, in this example, you can see that even though a large part of the individual features of each letter are missing, people have no trouble reading the word perception. So here is an example of perception going beyond what is out there. Even though no particular letter is written in this word, we can make inferences about how the organization of what the stimuli is could be depicting the individual letters and then the word perception. So the context of the word perception helps us perceive those letters as well. The visual system allows us to transform light into psychological experience. In fact, it's designed so as to project a sharp image of the world outside onto the back of the eyeball on the tissue known as retina. Retina is a light-sensitive tissue and has many cells, about 200 million photoreceptors, cells that receive light and have the capacity to transduce it into electrical signals that can be sent to the brain. Light enters the eyeball through the pupil, which is a tiny hole in the iris. The refractive properties of the cornea and the lens allow for the sharp image to be projected onto the back of the eye, where the retina is. The retina has two types of photoreceptors, rods and cones. Both are differentially sensitive to light and carry out phototransduction. If you look here in the figure on the right, you can see that they are named after their structure. Rods have cylindrical ends, whereas cones have conical ends. The photochemicals or photopigments are located in these ends at the back of the eye. Having these two types of photoreceptors or cells that respond to light under different conditions affords us dual functionality. They are also distributed differently on the retina, so you see many more cones in the fovea. Fovea is the part of the eye where the light falls directly when you're looking straight ahead or from straight ahead. And that's where most of the cones are. Conversely, rods are much more prevalent in the parafoveal areas. In general, we have very few cones in the peripheral retina, and most of the cones are concentrated in the foveal retina, whereas rods are much more in the periphery. Now notice that even when I say very few cones, I'm talking about 10,000 per square millimeter, so there's plenty of cones on that side. So what does this differential distribution and their differential ability to transmute light do for us? Cones have a photopigment called scotopsin, which is sensitive to lower wavelengths of light and are wired together to give us high acuity. So they can differentiate between points that are close apart much more effectively. We also have three types of cones, so that means that these are able to distinguish color. Each type of cone is sensitive to a slightly different wavelength of light. The small, medium, and long cones that are specialized for small wavelength of light are then specialized for blue color, and so on. Rods, on the other hand, are found, like I said, in the periphery. Their pigment is sensitive to a higher wavelength of light, and they are wired together to give the system much more sensitivity and less acuity. I'll talk about that in a minute. So how do we know all this? Much of what we understand about the visual system comes from a technique called single unit recording. Here's an image of some visual neurons. Single unit recording involves putting an electrode close to the cell body of a neuron and recording its action potentials as the cell fires. The frequency of action potential is recorded as various kinds of visual stimuli are presented to the subject. This leads scientists to conclude the extent to which that cell is involved in processing that stimulus and how the function of that cell correlates with both the changes in the stimulus as well as the changes in the perceptual decisions of an individual. So very frequently, using this method, scientists will map out the receptive field of any given cell within the system. The receptive field of the cell is the area of receptors, the space or body that affects the firing rate of that given neuron. So for example, a cell that is in the back of the retina, in the fovea, responds to light in the straight ahead. So the receptive field of this cell B is straight ahead. Receptive field of the cell A then becomes a space that is straight up from the fixation point and from where light, when it falls on the cell, induces activity in that cell. So recording from axons coming from the different cells in different parts of the retina has led to our understanding of how the retina represents space. It's important to realize that the optic nerve is not just a nerve tract that leaves the eyeball and carries information to various sites in the brain, or that the retina is simply made up of photoreceptor cells. Instead, the retina has a rich complex of neuronal cells. The photoreceptor cells, for example, are connected to the ganglion cells, whose axons make the optic nerve. And between the photoreceptor cells and the ganglion cells, there are other connections, both vertical and horizontal, that make the retina capable of integrating and processing to some extent the information coming in before it sends it to the brain through the optic nerve. This then makes the retina capable of edge enhancement. So retinal cells are able to enhance edges across bright and dark spots. Lateral inhibition leads to the ganglion cells having receptive fields that with a center surround organization. Let's try to understand this a little bit and see how it leads to edge enhancement. Here's an example neural circuit. Imagine that cells 1 through 7 are photoreceptors that are connected to this ganglion cell B via these excitatory connections. Some of them are connected directly to the ganglion cells. Others are connected through interneurons or other horizontal connections within the retina. What that does is that if any of these photoreceptor cells are excited, then cell B has a chance of being excited. Now, if this relationship was such that 1 unit of excitation on any one cell led to 1 unit of firing rate in cell B, then as you increase the number of photoreceptors that are excited, you get more and more firing in this ganglion cell B. If only cell 4 is excited, you get 1 unit of activity. If cells 3 to 5 are excited, you get 3 units of activity. If cells 2 to 6 are excited, you get 5 units of activity, and so on. So this is what happens when these are all excitatory connections. Conversely, if it were the case that some of these connections, more specifically the surrounding connections, were inhibitory, then what we see is that only cells 4, 3, and 5 result in excitation in cell B, and every time the surround is excited, we have some inhibitory integration. What that does is that when cell 4 is excited, you get 1 unit of activity. If cells 3 to 5 are excited, you get 3 units of activity. But when cells 2 through 6 are excited, then that activity shows subtraction, and it is now only 2 units. So 3 units of excitation and 2 units of inhibition leads to 2 units of firing. And when cells 1 through 7 are excited, you actually see an overall inhibition of the ganglion cell B. So this centers around antagonism between excitation and inhibition, then leads to tuning of responses for ganglion cells. So the ganglion cell B has a highest response only when the excitatory area is stimulated. It has its lowest response when only the inhibitory area is stimulated, and an integrated, summated intermediate response when both the excitatory and the inhibitory areas are stimulated. That leads to contrast enhancement or increases in the difference of activity across edges of stimuli. So we can try and work through how different cells across an edge might respond due to this lateral inhibition. If you look at cell B, it has the same brightness as cell D, and it's getting the same amount of excitation from the light and the same amount of inhibition from its neighbors. So cell B's activity is the same as cell A and cell C. Cell D, which is on the edge, however, gets less inhibition from its neighbors in the dark. these cells are not only themselves less excited, but they're also then sending less inhibition to their neighbors. And because there is less excitation in cells E, F, G, and H, you see a lower activity in these cells. And this also results in a higher activity in D cell due to this lower inhibition. Similarly, for cell E then, because it's getting more inhibition from cell D than cells F, G, and H from their neighbors, it ends up having a lower activity than cells that are in the same dark patch of that stimulus. That is what I mean by enhancement of edges. So the contrast of activity between the cells that are on the edge is higher. This actually leads to a perceptual contrast. And you can see in this stimulus, for example, even though the gray bars have the same intensity from one edge to the other, in each bar, we tend to have a slightly darker percept on the edge that's abutting the lighter side and a slightly lighter percept on the edge that's abutting the darker side. And for different people, you see this illusion being more prominent in one or the other of these bars, and you might find yourself seeing edge enhancement, more edge enhancement here or here compared to in the lighter phases or the darker phases. But the point is, we can see this illusion, or we can actually demonstrate this contrast enhancement through stimuli like these, called the MAC bands. This is a figure from your textbook. What I would like you to do is kind of work your way through how a stimulus like this may be perceived based on what we've learned about lateral inhibition and contrast enhancement. So now that you know how center surround receptive fields are organized for ganglion cells, for both rods and cones through these lateral connections within the retina, it will be easy for you to understand why this causes the cone system to have better spatial acuity and the rod system to be more sensitive to dim light. This is simply a result of greater spatial summation in the rod system. In this example here, you can see that if more rod photoreceptors are sending excitatory signals to the ganglion cells, they can then not tell these two spots apart as easily as a cone system where different ganglion cells are excited by these two spots of lights through less spatial summation. Conversely, this results in more response in a ganglion cells connected to a whole bunch of rods when there's dim light, whereas ganglion cells connected to cones do not show a response. So having the rods and the cone system allows us to have both acuity and sensitivity in dim light conditions. Here's a nice demonstration of lateral inhibition. If you stare at this stimulus somewhere here where the cursor is, hold your fixation for a good 20-30 seconds. As you stare at the stimulus, what's happening is that parts of the stimulus which are white are stimulating photoreceptors and ganglion cells in that part of your retina. Now you have to maintain fixation to do that, and that gets super stimulated, and the parts that are dark are not exciting those photoreceptors, that part of your retina. When I turn the stimulus off, what happens because of lateral inhibition is that you see brightness in the dark parts and darkness or gray in the light parts. And this image is inverted. Did you see something interesting here? All right, moving on. So then, going from the eye to the back of the brain in the visual cortex, the primary visual cortex, are these nicely processed and integrated signals. The first stop is at the thalamus, which is the sensory relay station for the brain in the lateral geniculate nucleus, and then from there to the back in the occipital cortex to the primary visual areas, also known as the striate cortex. These areas are organized much like the retina, with cells which are doing much the same thing, that is, edge detection. The only difference is that in the primary visual cortex, we are now summating receptive fields of cells like the retinal ganglion cells and the LGN cells, which are centers around, more circular in organization, across space. So when we summate a few of them that are lined up from left to right or right to left or top to bottom, what we end up with in the primary visual cortex are cells with receptive fields that prefer a bar or a line of a particular orientation. So we are now going from a small spot enhancement to our edge, a real edge enhancement. Within the primary visual cortex, there are many neurons with different receptive fields focusing on different locations in the retina and parallelly processing all the bars and edges of all the stimuli that you are seeing at any given point of time. And these cells, once again, have been recorded from, and you can see that a cell that prefers a vertical bar of light shows tuning to that orientation. So the largest response is for that orientation, and then the response kind of decreases or dies down in other orientations. That's tuning. David Hubel and Thorsten Wiesel actually won a Nobel Prize in medicine back in '81 for their work in understanding how neurons in the primary visual cortex represent the world. And they showed that amongst the different layers of the primary visual cortex are cells that are orientation detectors, cells that respond to color, and other complex cells that not only respond to orientation, but also to a stimulus of a certain size, so or movement. So everything is being processed in parallel in the primary visual cortex, which is then parceled off to many other areas in the brain for further processing. So the primary visual cortex starts processing different features or aspects of the stimulus, and further areas in the visual cortex going into both the inferotemporal lobe and the posterior parietal lobe, you can see here, are pathways of processing for different aspects of the stimulus. The ventral or the temporal pathway focuses largely on object recognition, so what it is that we are looking at, and the dorsal stream going dorsally to the posterior parietal cortex cares much more about movement, spatial relationships, and actions that result from spatial integration. To get an idea of what's going on in these many different modules of visual processing in the cortex, let's watch this movie and see how these pieces of information are then put back together into our percepts as a whole.
Our eyes may be the front doors to vision, but strangely enough, they aren't where sight happens. It starts when incoming light is converted to electricity by these strange devices called rods and cones. Kiki, whom your eyes are gathering now, will help us with the rest of the trip. First, incoming light information is flipped upside down. Next, the information breaks up into fragments. Then, at the crossroads of the visual fields, it splits. Sorry, Kiki. The fragmented pieces then make their way to the primary visual cortex, found at the back of the brain. This layered region filters and codes the information. Now Kiki's trip gets even more complicated. All that coded information is parceled out to as many as 32 locations for finer processing. The cerebral cortex, the brain's quarter-inch thick thinking cap, consists of tightly packed columns of nerve cells. It's the number of columns and their complex web of neurons that set us apart from other animals, what makes us human, in fact. Here, we finally see the world. Inside each column, neurons form networks of fierce activity. Information continually bombards the brain. To manage it, the cortex uses the division of labor principle. Here, neurons begin sorting the still jumbled scene. We know Kiki is in there somewhere. Every column has a complete set of neurons, each designed to seek out a specific angle from 0 to 180 degrees that might be found in the various shapes within the scene. It's a bit like finding the edges of a jigsaw puzzle first. As the process continues, thousands of lightning-fast tasks rage beneath the brain's surface. It's a race to make the round edges sharp, to find in the incomprehensible jumble something or someone we recognize. Eventually, the outlines of a face appear. All of these visual bits and pieces are processed and coded by assigned neurons. While one set of neurons is busy reading the contour of a cheekbone, another will read the shape of the eyes or the slant of the nose. It's an incredibly evolved bit of natural computing, and at the end of this split-second journey, when everything is reassembled, a familiar image appears. So many combinations. Experts speculate that the brain builds on what it learns while processing simple patterns, enabling it to perceive increasingly complex shapes or other forms of information.